Thank you all for joining me today. My name is Ian Taylor and I'm a director here at FlowJo. Go ahead and kill my video now and we can get into the presentation. Today's presentation is going to be on GeneSet enrichment analysis. If you have any questions afterward, I would encourage you to write to me, taylor@flowjo.com. I'd be happy to troubleshoot with you any issues that you might be having. We could even set up a one-on-one demo to go over your data and obviously any questions associated with this particular platform. As mentioned previously, this video will be placed on the FlowJo University website so that you can view it later, as well as the deck of slides that I'm using today. And without further hesitation, let's jump into GeneSet enrichment analysis. So just to get started, what is GeneSet enrichment analysis and why might you want to perform it? GeneSet enrichment analysis essentially compares a gene set of interest with a set of gene sets in what we would call a GeneSet library. This information is used to infer a biological state or some kind of biological information about the gene set being tested. And we utilize a Fisher's exact test, which is a statistical test to tell us how closely the gene set of interest matches various different gene sets in the GeneSet library. In order to access this platform in SeatGeek, you're just going to right-click on a gene set at the top of the workspace and choose Enrichment Test. That's going to bring up this window. And in that window, you'll see that you have to select a gene model. We'll discuss a little bit more about what that is, but in this case, I've chosen the sample itself. You'll have to choose a GeneSet library. Here I'm utilizing the B-cell GeneSet library that we actually developed ourselves here at FlowJo. And you'll have to set a p-value cutoff. Now that p-value is referencing the Fisher's exact test, and it's noting which gene sets will be included in the test results. You'll also notice in the background that I've done some differential expression analysis, and that has given me a gene set on which to run this test. So again, a model must be selected in order to run the GeneSet enrichment analysis, and that model is typically going to be the sample that you've loaded, and that's usually a good enough model for the test to run. Ideally, this would be the library of genes that were used for sequencing and specifically for gene calls in your upstream of this analysis. But again, the raw sample will work pretty well for that. Next, you will need gene sets because you're going to select a gene set in order to run GeneSet enrichment analysis. And there are many ways to set gene sets within SeatGeek. Probably the most popular is to use an automated approach. For example, Seurat will produce differentially expressed gene sets for each cluster that it calls. You can also do differential expression analysis, as I showed in the previous screen, in order to identify potential genes of interest for a given state or population. So here we're showing the outputs from Seurat that are, I guess, most graphically pleasing. In the plots on the left, we see gene coverage per cluster that's been identified. And in the plot on the right, we see t-SNE dimensionality reduction parameters that were developed by the Seurat pipeline, as well as the clusters produced by graph-based clustering using a k-nearest neighbors algorithm. And that's been color mapped by those clusters, so we can see them very clearly in the plot. What we don't see in this view that is available in the workspace is a collection of gene sets, one per cluster, which are the differentially expressed genes defining those populations. So these are perfect candidates on which to run the GeneSet enrichment analysis on. Great. Again, in order to run GeneSet enrichment, you're simply going to right-click on a gene set, choose Enrichment Test, and in the window that pops up, shown at the bottom of this screen, you're going to select your model, your GeneSet library, and some p-value cutoff for Fisher's exact test. The results from that test, once you click Run, are going to appear in a new window, the information in which can be exported as a CSV file in case you want to take that into Excel or put it somewhere else, use it for further analysis perhaps. That information is the gene set that's been called. In this case, we have located a potential naive B cell subset identified by this upregulated naive B cell gene set that we produced in a different analysis. And it's given an associated p-value, which is determined by the nature of the data there. We see that there are 4 genes in the Seurat cluster 1 gene set that are in common with the naive B cell upregulated gene set. So that's a good enough match that it gives it a pretty significant p-value, and that would probably give me enough confidence to call this cluster potentially a naive B cell subset. So phenotype or cell calling is one application of many that can be performed with GeneSet enrichment analysis. Also of note, and really useful for this platform, you can create your own GeneSet libraries. And in order to do that, you're going to select a couple of gene sets. Here you can see in the background in the workspace, I've selected down B cells and upregulated B cell gene sets. And that's just a generic set of gene sets in this case. In the Genes tab, I'm then selecting Export, and I'm given the choice. I can either export CSV gene sets that will be imported just like you see these gene sets here, so I could take them to a new workspace, or I can select to export a gene matrix transpose file, or GMT, which is the popular way of storing and utilizing gene set libraries. So this GMT file is what we would consider actually a gene set library file. These are available from many different databases such as Go or GSEA that you can use and import directly into SeatGeek. Cool. Now, when setting up a gene set library, there are some really important considerations that you should be aware of. For example, you want your gene sets within the library to be at the same level of biological inquiry. So you want to be using the same model organism. You want these gene sets to come from the same tissue type. You want them to specify the same level of heterogeneity that you're testing against. So if you're looking at B cells only, you probably want gene sets that are in your library that are derived only from B cells, if we were trying to identify memory, plasma, and naive, for example. The classes of comparisons should be the same. So you want to be looking at similar comparisons, and you want a roughly equivalent depth of gene coverage. So if I have one gene set that describes my naive B cells and it's thousands of genes long, then I don't want my memory gene set to only be a handful of genes. That would be an inappropriate depth of coverage for memory B cells in that case, relative to our naive B cells. In this example, we are doing differential expression analysis, which is oftentimes the precursor to gene set enrichment analysis. And specifically, we have a data set that comes as demo data in our latest release, which is lupus data provided by the AMP consortium. And that data contains both, it's kidney data, and it contains both disease state cells and those from healthy patients, so that you have something to compare against. And of course, using differential expression analysis, we can find the genes that are upregulated in the disease state versus the control. And interestingly enough, of course, as a validation, we want to see if that gene set will actually enrich to something meaningful. And sure enough, when we run it on the publicly available KEGG diseases for humans gene matrix transpose, the #1 hit is for SLE or lupus at an extremely high significance value. So we're trusting that the gene set enrichment analysis is working correctly, and it's pointing to something significant. Now, this may not be a part of your discovery workflow. This would be more of, again, a validation step. But if you had pathways that you were interested in exploring within the lupus disease state, you could run a gene set enrichment analysis specifically for pathway enumeration and further discovery. And that's about all the slides I have on GeneSet enrichment analysis. Just want to note a few different resources that you can check online if you want to do some self-help and try and understand better the GeneSet enrichment analysis platform. There's also support available, SeatGeek@flowjo.com. You can always write to me, Taylor@flowjo.com as well. That's TAYLOR@flowjo.com. Be happy to set up a demo or simply to troubleshoot whatever issues you might be having. Might also mention the Exchange site where you can download plugins such as Seurat and Monocle, which do unbiased clustering and lead you to differentially expressed gene sets very quickly. Again, that's at exchange.flowjo.com. And of course, if you haven't done so already, I would strongly recommend that you download SeatGeek and potentially sign up for a free trial by following the instructions at this URL. For the last bit of this demo, I would just like to walk through some GeneSet enrichment analysis with you live here. So I'll leave these links open in case anybody's still writing those down. Now would be a great time. If you have questions, feel free to post them into chat or save them and send me an email afterwards. Here we see some B cells that have been pre-analyzed by myself. These B cells come from 10X as part of a set of demo data that they provide. So it's a very homogeneous population, but we were able to actually do some clustering in Monocle, and we got some good results there. So I'm going to just illustrate that here. Actually, a better example before cell calling. Yeah, quality genes. So here we see the dimensionality reduction that's produced by the Monocle tool, which is developed and maintained by the Cole Trapnell lab, wherein pseudotime is predicted for each of these cells, which gives us states of differentiation according to differentiation trajectories. So it's thought that the cells in the center of this tree are generally your basal state, or those cells that are least differentiated from the rest. And as you move outward in any of the directions of these branches, you're getting more and more differentiated cells that are progressing towards some final state. You'll notice that these two plots look very similar. I'm just illustrating two different ways that you can show these parameters. In this case, I'm color mapping by the Monocle state parameter, which is generated by the plugin. And in the plot on the right, I'm illustrating an overlay on those same set of parameters, illustrating each of the states or clusters that have been predicted by the Monocle plugin. And I've actually just denoted in color the different branches that are coming from that differentiation, projection, and prediction by this R package. And again, this is a plugin that has some dependencies in R, just because that's the platform that the Cole Trapnell lab chose to use for, and is a popular tool for designing these bioinformatics algorithms. But it's made very simple and easy to use in SeatGeek. I might just illustrate that if we select any population, go to the Workspace tab and look under Plugins. I've already set up my Monocle plugin, so it's available here. When you first download the plugin, it should have some instructions that tell you exactly how you can set this up and get it running on your machine. Any problems there, feel free to contact me, and I'd be happy to troubleshoot again. So getting into GeneSet enrichment. Before we do GeneSet enrichment, we're going to need two things: a set of differentially expressed genes, as well as a library that we want to compare those genes, that gene set, against. In this case, we have a Monocle state 6. Looks like it has a lot of differentially expressed genes. How did we derive this gene set? Well, that's using differential expression analysis, which I covered yesterday in a similar webinar, but I'll go through that very quickly now. So if I open up my quality control population, removing the mitochondrial high cells, so those that might be dying or dead cells. I switch to gene view here, which is what we're currently looking at. And if I'm interested specifically in Monocle state 6, I'm going to place my comparator population on the x-axis. and that is going to be all those cells that are not in monocle state 6, and my test population, which is monocle state 6, right here on the y-axis. From here, in order to run the differential expression analysis, we want to open a volcano plot. You can click on this button here, and you can see I've already drawn a gate that is in the positive fold change region with significant adjusted p-values here on the y-axis. So anything above about 0.05 or 5% significance value. And I'm going to maybe just make a little adjustment here so we can see that the number of genes in this gene set is adjusting live as we change that gate or gene set definition. So now that we're down to 248 of the most significant upregulated genes in the monocle state 6, I'm going to go ahead and run gene set enrichment analysis. To do so, I just right-click on this gene set, choose enrichment test, and that brings me to this window where I can choose my gene model. In this case, I'm just using the raw sample here because I don't actually have the list of genes used for library construction at the beginning of this sequencing of this B-cell data set, but this should work just fine. For GeneSet library, I know that in my downloads folder, lots of things, but one of which is the GeneSet library we developed in-house for B cells. Now, the development or curation of GeneSet libraries is something that you actually may want to perform yourself. You can see we've done a number of different phenotypes. In PBMCs, we've actually divided these up into their various constituents. So for B cells, we have our memory, naive, and plasma gene sets, as well as an unknown gene set for all those B cells that were not identified by immunophenotyping in this case. And I'm going to go ahead and click that so that we've loaded our B cell gene set library. I'm interested in seeing any degree of significance with regard to our Fisher's exact test. So I'm going to go ahead and set this p-value to a very insignificant level, and it will show me anything above a 0.9 or below a 0.9 in terms of p-value, above a 0.9 in terms of significance. So if I click run, we're going to get the results here. And you can see that indeed for this particular cluster, nothing very significant came up. But we were able to make an estimation that there could be B cells here, or these could be naive, that there could be plasma cells here, or these could be naive B cells. In this case, because of the high p-value or low significance involved, I might just say this is an unknown cluster because, again, the p-value is just not significant enough to call this one way or the other. However, we do see that there are 3 genes in common. We won't know exactly what those genes are unless we do some kind of Boolean logic. I happen to have the gene sets associated with this gene set library loaded into this workspace, so we might just check that. If I select this monocle state 6 upregulated gene set, and was it plasma? Just control click to select both. I can go to the genes tab and choose the intersection. And we see that there are a number of genes that are in common between these 2 gene sets. And if I double click on that gene set, we can see exactly which genes were found to be in common between my monocle cluster number 6, that's here, or sorry, right here, and the plasma B cell gene set we developed in order to construct a gene set library. And so you might go and look in the literature or check gene cards or whatever you prefer to use for exploring these different genes and get some idea of what might be going on that's similar in plasma cells with regard to the cluster 6 in monocle. I could also note that cluster 6 is this dark red guy, so that's the branch that's pointing out this way. So you can see this is a really versatile tool. And really, gene set enrichment analysis is the final piece in most analyses to tie in some biology to, or deeper biology to the analysis that you're performing. And so it's really important to get that included into the SeqGeek platform. Now that it's here, we hope we'll get lots of great feedback, even constructive criticism, to allow us some insight into how this can be best suited to the needs of researchers. Again, if you guys have any questions or concerns, I would strongly recommend that you reach out to SeatGeek@flojo.com or to myself, Taylor@flojo.com, and let's see what SeatGeek can bring to your analysis. Thanks again for joining. We'll see you later.